Hello everyone. All right, I'm recording this quick video to go over mutations. I couldn't really find a decent video that I liked, and I also know that you all have the same sheet. So first off, you need to kind of make a note or notice what your original DNA looks like, and then notice what the original mRNA is supposed to be, and then what the amino acids are supposed to be. So that's going to be helpful so that we know what we're supposed to be looking at before we start looking at the mutations. Okay, so we're really going to go over 4 types of mutations, but there's only 3 on the sheet, although the 4th one's very easy. All right, so our first one is a substitution. So looking at our mutated DNA, which letter, which nucleotide seemed to change? So take a moment, take a look, see if you can figure out the difference between our normal and our mutated DNA. All right, so hopefully you could tell that it is the letter T. The letter T and C, they swapped. So we were supposed to have C, but instead we had T. So instead of having CTT, GAA, now we have TTT, AAA. And that changed our amino acid from glutamine to lysine. So that is a substitution. Only one amino acid changes. All right, and our next one, this is a very bad or advanced mutation because pretty much the entire protein changes. Instead of changing one amino acid, we change a bunch. And so take a look at our inserted DNA, insertion mutation. So you can figure out which of these letters changed, which nucleotide changed. All right, so hopefully you can tell that everything is the same except we added a letter. A C was added. So we are supposed to have CTT, CTC, CTT, CTC. But instead we inserted a letter, we added a C, so now we have CCT, TCT. And if we kept going, we would see that all of the reading frames changed. Because remember, the ribosome reads everything in codons, or sets of 3 letters. And so every codon after this mutation has now changed. And therefore, all of the amino acids after that mutation will now change. This is a very bad mutation to have. Most likely it's going to result in a non-functioning protein, and therefore it probably will cause death or will be like a really short-lived life. Substitution, there are several diseases that we know of that's caused by a substitution. One of them being sickle cell anemia, another one being breast cancer, and so usually those are just slightly off, Insertion and deletion are really bad. The whole protein is just messed up. So we call this a frame shift mutation. An insertion frame shift mutation. And I should add, we call substitution also a point because there's only one point in the DNA. Point mutation. All right, our last one is a deletion. So in this setup, we've deleted a letter. So what letter did we delete? Take a look at your original DNA and your mutated DNA. What's the difference? So after watching and looking, the letter we deleted was an A. We took that A right on out. We should have had AAA and then ATG, CTT, CTC. Nope. We now have TGC, TTC, TCC. So every point after this deletion, every amino acid has changed. Again, it's a frame shift mutation. It's probably going to be lethal or a non-functioning protein that can be really problematic. And if it's not problematic, then it's probably really superficial, like red hair color or blue or green eyes. All right, our last mutation is a substitution. So think about this codon chart, all right? There's only 20 amino acids, but if you do the math, 4 letters, right? AGUC, and 3 spots, because each codon's 3. So that's 64 codons. But there's only 20 amino acids. So look at alanine. Alanine has 4 different codons that result in the same amino acid. GCU, GCC, GCA, and GCG. All will give us alanine. So a silent mutation is one in which DNA nucleotide changed, but after transcription and translation, no amino acid changed. It gave us the same one. So if that's the case, we call it a silent mutation. So that's all I have for this video. Next up are a couple practice problems from Khan Academy. And then we have a practice assessment because we have our test on Wednesday or on Thursday next week. Take care and good luck studying.